So good morning, everyone. I like to make these very informal. So if you have any questions at any moment, just feel free to interrupt me and ask. I really don't mind being interrupted. Actually, it's kind of nice to get questions as you are talking. Okay, so I usually like to start my lectures like this by quoting one of my favorite scientists, Sidney Brenner. How many people have heard of him? Maybe he's not so well known in the engineering field, but he's kind of a really renowned person in biology, and he won the Nobel Prize in 2002 for his work in really understanding the genetic underpinnings of development, particularly nervous system development, and he did that using C. elegans as a model system. And he had these quotes that, have become very popular over time. And this is one of my favorite ones. And he once said that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And so he really, with this quote, put a lot of emphasis in development of new technologies, right? New technologies really propel forward new biology. And if we really take that quote to heart, the last 2 decades or so have been an amazing time to be doing science because we've had really a lot of technological revolutions in many different fields, in light microscopy, in electron microscopy, sequencing techniques. These tools have really undergone revolution thanks to development of new detectors, new sort of methodologies. And today I'm going to focus on the revolution that has happened in light microscopy and tell you about how that revolution has helped us almost change sort of our cartoon textbook models of how we think about our genome and how that genome is folded and organized inside a nucleus. So before I get into that, just a brief overview about my lab here at Penn. We like to study how cells are organized in space and time. As you probably know, cells are not just a soup of proteins and nucleic acids. They're very highly spatially compartmentalized and organized. And my lab really likes to be able to visualize that spatial organization of proteins, DNA, and understand its functional relevance for the cell. And we study this in the context of 2 main biological problems. On one hand, we're interested in how organelles like mitochondria are spatially organized within the cell and how they get to where they need inside the cell, and the role of the microtubule cytoskeleton and motor proteins that mediate their transport from one location to another and their positioning and how that breaks down in diseases. And there's quite a lot of interesting work going on in the cytoskeleton field, also in the CEM bit, and probably you'll get to hear about that in some of the lectures. But we're also interested in the organization of the nucleus and how particularly the DNA is spatially organized within the nucleus and how that organization impacts gene activity. And as we work on these topics, we often, again, realize that existing methods have limitations. And so we try to develop new methods to overcome these limitations. And I think I will touch upon some of those methods in my talk. So today I'm going to focus on DNA, on chromatin, and on the nucleus. And as you know, you're probably familiar with the Human Genome Project. This was a very ambitious project that aimed to sequence the entire human genome, and it was completed in 2003. It generated an assembled sequence of the human genome, along with many other species, including C. elegans, mouse, etc. And this was really a very impactful project because many diseases are associated with genetic mutations, right? Mutations to the linear code of the DNA. And this really helps us understand those diseases and potentially find cures for them. But, the Human Genome Project is not the end of the story, because we also have many diseases that don't have any genetic underpinnings. There are no known mutations that cause those diseases. So they come from things that are beyond just the linear sequence of the DNA. And today we understand also that the linear sequence of DNA is important, but there are factors that are beyond the linear code of the DNA that are also important, so-called epigenetic factors. These can be chemical modifications to the DNA or to the proteins that are associated with the DNA that change whether a gene is on or off and can regulate gene activity. And these sort of epigenetic factors, in addition to the sequence of the DNA, help regulate gene function. Part of that epigenetic regulation is how the DNA is folded and organized in the space of the nucleus. If you ever, I don't know, have thought about this, but if you take all our chromosomes and stretch them and put them end to end, the length of that is 2 meters. So that's, longer than most humans. So this is really a huge biopolymer. And that biopolymer needs to fit inside a nucleus. The nucleus is about 20 microns in diameter. So imagine taking 2 meters long rope and fitting it inside a 20 micron space. That requires an enormous amount of folding, compaction. And again, that folding and compaction we now understand regulates the activity of the genes, whether they're on or off, how they interact, how a gene interacts with its regulatory sequences. What I'm going to do is try to first show you a video that shows our textbook model understanding of how that 2 meter long DNA becomes folded and compacted inside the space of the nucleus. And then we will go through parts of that textbook model, how we got to understand it, and some of the new technologies that are challenging it. Okay, so here what you see is, the double helix structure of the DNA. And if I play this movie, it's just sort of presumably in the nucleus jiggling around, and it says this is like a 100,000 magnification. And then you will see some proteins, they come in out of nowhere and they just magically land on the DNA, and more proteins come. And the DNA makes these turns around these proteins. And it actually turns in almost 2 turns around these proteins to form the nucleosome. Nucleosome is this packaging unit of chromatin, and you get several nucleosomes. And these nucleosomes assemble together and compact together into what is known as a 30 nanometer chromatin fiber. And then that chromatin fiber further folds. This wrapping of the DNA around the histones to form the nucleosome, and the compaction of those nucleosomes is what takes this 2 meter long biopolymer and really shrinks it and compacts it so that it can fit into the space of the nucleus. And here's that textbook model again, more in static form. So you have the linear, sequence of the DNA, and these are the length scales. So this is in the 10 to 20 angstroms range, and then you form the nucleosomes by wrapping the DNA around these proteins called histone proteins. That's more in the 10 nanometer range. Multiple nucleosomes then compact together into a 30 nanometer fiber, which further folds. And the most compacted form is the chromosomes that you see in mitosis when a cell is dividing. And so why do we really care about this? again, the conventional wisdom is that if the DNA is in the form of these 30 nanometer fibers inside the nucleus, that's a very compact structure, right? And the code of the DNA is kind of hidden in that compact structure. And so things like polymerases that need to read the code and translate it into, transcribe it into RNA, they cannot access the genes that they need to transcribe. And so those genes are inaccessible, and so they're silenced, right? So in order for a gene to become activated, the chromatin must unwind into more like a 10 nanometer fiber form so that the polymerase can bind and read the sequence of the DNA and transcribe it into RNA. So we care because we think that this folding and compaction really regulates which genes are on and off. And further, if you look at this larger length scales in the 100 nanometer range, where you have that sort of further folding of the DNA, we know that genes, their activity is also controlled by regulatory sequences in the code. So you have a gene, and you have what is known as an enhancer. The enhancer contains all the polymerases and the transcriptional machinery that turns the gene on. And that transcriptional machinery needs to be transferred to the gene. But often when we read the linear sequence, the gene and its enhancer are very far apart in the linear sequence. They can be megabases apart. So how do you transfer all the transcriptional machinery that's on the enhancer to the promoter of the gene? You do it by this higher order folding, bringing in the 3D space of the nucleus, sequences that are far apart into close physical proximity. And so this, again, compaction folding is supposed to control the turning on and off of the genes. So it's really important that we understand how this looks like inside a nucleus and how a gene is, regulated and its activity is regulated. So this is, this is a textbook model. How did we come up with this textbook model, right? It's just a cartoon. And in fact, there is no microscope on Earth that can generate that movie that I showed you in the previous slide, right? There's no microscope that can actually capture the folding of the DNA at that level of spatial resolution and with that level of temporal resolution. So our textbook model understandings in biology usually come from bits and pieces of evidence that we piece together. In this case, those bits and pieces of evidence come from chromatin that has been reconstituted in vitro. So basically, you take a piece of DNA, you mix it with the histone proteins, and it self-assembles into chromatin. And then you can use an electron microscope. Electron microscope has very high spatial resolution because you're using electrons that have very short wavelengths. And you can make pictures, right? So this is an example of the 10 nanometer fiber. You can beautifully see that beads on a string, the pearl necklace type organization, where each one of these beads is a nucleosome, and then you have the linker DNA in between. And so that's, the 10 nanometer fiber in the textbook model. And then under certain, if you play around with the salt concentration in your solution and you add additional histones called linker histones that are supposed to, link multiple nucleosomes together to further compact it, then you get something like this. And this looks more like the 30 nanometer fiber in the textbook model, where you have multiple nucleosomes compacted together. And in fact, people have done much higher resolution imaging of this and have shown that the nucleosomes really stuck together in a very particular manner. And specific nucleosomes contact each other, forming a very regular, very beautiful pattern within that 30 nanometer fiber. But of course, these were really landmark studies that told us about, how chromatin is assembled, but they are in vitro reconstituted models, right? So they're not chromatin inside a nucleus. So ideally, we want to see what that looks like inside a real intact nucleus. And you can do the same thing. You can use electron microscopy to visualize cells and their nuclei. And this is another example of an electron micrograph. And this is the nucleus, the round structure. There are things called nucleoli. Inside the nucleus, this is RNA processing center of the nucleus. And then you see these dark and light patterns inside the nucleus. And those dark and light patterns are chromatin. And you can see some regions that are much darker, like at the edge of the nucleus, right? You see these very dark regions. Also around the nucleoli, there's accumulation of dark regions. Those dark electron-dense regions are heterochromatin. So presumably, the more dense, more compact, those 30 nanometer fiber-like structures, because they're very compacted, they appear dark in the electron micrograph. And then the more light regions are the more open regions of the chromatin, more presumably transcriptionally active, more expressed genes are in these euchromatin regions of the genome. So these type of images allow us, again, to sort of map chromatin into different domains, like the heterochromatin and euchromatin, the compact and the open chromatin. So we know that these compact and open regions exist inside a nucleus. But the issue is that we can't really see what the structure of the chromatin looks like within these compact and open regions. So we don't really, can't zoom into here and see those beautiful 30 nanometer fibers. And the reason for that is because electron microscopy has low contrast. So nucleus is not just chromatin, right? There are many other proteins and nucleic acids inside the nucleus. And so it becomes hard to really discriminate what is chromatin, what is DNA from everything else. The contrast is low. And we don't get, that high resolution that you get in vitro where you only have chromatin and nothing else. Does that make sense so far? Any questions? And so, our understanding of sort of chromatin for a long time was, these in vitro reconstituted fibers, and then the idea that those assemble into a heterochromatin and euchromatin compartment inside the nucleus. Now, I told you electron microscopy has low contrast, so it doesn't allow us to really visualize with high, confidence these different structures inside an intact nucleus. What about light microscopy? So light microscopy has complementary advantages to electron microscopy. In light microscopy, what we usually do is we use labels. These are fluorescent labels, so fluorescent molecules that we attach to specific proteins, right? We can attach them to, for example, nucleosomes, so they light up. And this solves the contrast issue. Fluorescence microscopy has very high contrast because everything else is going to be dark, and you're only going to see the thing that you label. And you have molecular specificity. You know what you're labeling, right? Because you have put a label on it. You know that what you're visualizing is the nucleosome. So this is great because it solves a lot of the issues of electron microscopy. So how about we use light microscopy to visualize chromatin? And people have done this. You can put fluorescent tags on the histones to visualize them using light microscopy. And usually the images that you get look something like this. You see the beautiful shape of the nucleus, right? Everything else is dark. You see the background is very dark. You don't see anything else. It's just the nucleus and it's just the chromatin. But if you zoom into it, again, you don't see any structure, right? You actually see a big blur. There are some regions that are darker again, some regions that are lighter, kind of like in electron microscopy, but everything looks very blurry, and there is no structure in these images. This is because the spatial resolution of light microscope is much lower than an electron microscope, right? When we do light microscopy, we use visible light to take these images. And visible light has wavelength between 400 to about 800 nanometers. When we use visible light to take these images, because of that wavelength, things that are smaller than the wavelength of light cannot be visualized. I don't know if you've heard of the diffraction phenomenon. So whenever you have a wave, light is a wave, right? And it goes through an aperture. In this case, it's the aperture of your objective that has the lenses. It undergoes something called diffraction. This is the self-interference of light with itself. And that creates a diffraction pattern. So even if what you're visualizing is a point object, it doesn't appear as a point in your image. It gets blurred out into a larger spot due to that diffraction issue. And so because these length scales we're talking about are in the 10 nanometer range, and the wavelength of light is 400 to 800 nanometers, then really anything that's smaller than that becomes blurred out due to this diffraction problem. And I have actually a cartoon to demonstrate that. Usually the fluorescent tags that we're using can be very small. So typically we use fluorescent proteins like GFP, green fluorescent protein, red fluorescent protein. You might have heard of these. They're in the range of 3 to 5 nanometers in size. But if we try to make an image of that fluorescent protein on our microscope using light, the image is going to appear much larger, about 300 nanometers, and that's roughly half the wavelength of light. So the spatial resolution that you get is equivalent to about half the wavelength of light. Now, imagine that you have 2 of these fluorescent proteins. Each one is going to have an image that is given by that large blob, right? And when the 2 fluorescent proteins are very far apart from one another on your microscope, you'll see 2 blobs. You'll know there are 2 things there because you see 2 blobs. But if they get too close to each other, like in the 10 or 30 nanometer fiber, those 2 blobs will merge together. You will no longer be able to discriminate that you have 2 fluorescent proteins. They will appear as one. And this is what we mean when we talk about the resolution problem. It's your ability to discriminate 2 things that are in close proximity. And that, again, is limited by diffraction, by the wavelength of light.
Does the intensity of the light increase though, like if they overlap each other?
It does, but because each fluorescent protein, will give out a number of photons, which is kind of exponentially distributed, right? So some will be dimmer, others will be brighter. So again, it becomes very complicated to figure out how many just from the intensity, because the intensity is not one fixed value for each fluorescent protein. It's going to be a distribution, and that distribution is typically very broad and exponentially distributed. Does that make sense? Yeah, good question. Any other questions so far? Okay, so this resolution problem is not a new problem. It was known for a very long time. Maybe you heard of Ernst Abbe, a physicist. This is his tombstone in a city called Vienna in Germany. And if you zoom up to his tombstone, there's an equation there. And that equation is lambda, which is the wavelength of light, divided by 2n sine alpha. So this is where the diffraction problem comes from. This is telling you that the distance that you can resolve is given by wavelength divided by roughly, twice the refractive index of the medium. Sine alpha is the sort of angle of the lens that you have. Typically this comes out to be about half the wavelength of light. And so this, problem is so fundamental in physics that it's literally written on stone, on Ernst Abbe's tombstone. So it's no, surprise that for the longest time we thought this is it. We can't do any better using light. We can't reach a resolution that is better than roughly half the wavelength of light. But this is the revolution that happened in light microscopy, where this fundamental limit was broken. In 2014, the Nobel Prize in Chemistry was awarded to Eric Betzig, Stefan Hall, and W.E. Murner for their contributions to super-resolution microscopy. The Nobel left out somebody who had really greatly contributed, Xiaowei Zhuang, but she won the Breakthrough Prize in 2018 for her contributions to super-resolution microscopy. So this has been truly a revolution in light microscopy to overcome this diffraction limit. And I'm going to tell you about how you can do that. There are a couple of different methods that are out there, and I'm going to tell you about one common way where you can do that. So again, going back to that idea of having a single fluorescent protein, right, that gives out light, and that light in the camera of your microscope looks like a blob. Now, if you have only one fluorescent protein, even though the blob, the image is very large, you actually know that the fluorescent protein is at the center of that blob, right? And in fact, if we, talk more in mathematical terms, that image, that blob, is if you just look at the intensity profile of that blob, from one edge to the other edge, and you plot that, it's going to look like a Gaussian function. And you can pinpoint the center point of that Gaussian, right? You can fit it to a function, you can find the mean position, and you can do that with very high precision, much higher precision than this image itself. So in fact, the precision by which you can pinpoint the midpoint depends on the intensity, the number of photons emitted. The more photons, the sharper that Gaussian is going to look, and the more precisely you'll be able to pinpoint the center position. But we can actually do this with the fluorophores that we have with nanometer precision. Now, this is very useful, but it only works if you have one fluorophore, right? If you have two or more, just as we talked, their images are going to overlap, and you're not going to be able to discriminate one from the other, so you can't really find where they are in space. And so how do we overcome this problem? Now imagine that these fluorophores that you have are not constantly giving out photons, so they're not on all the time. Imagine that you have some ability to switch them on and off. And so imagine now you have two fluorophores there, but one of them is off. It's in a dark state, so you don't see it. You only see one, and then you can pinpoint its center position. And then you switch that one off, and you switch another one on that is nearby. And now it's shifted, the pattern shifted slightly, you can find the center position, and now you have broken the diffraction limit, right? You have pinpointed two things that are closer than the diffraction, you know exactly where they are. And so you're able to resolve them. Does that make sense to everyone? So this is how super resolution microscopy works. So the idea is that when you label your biological structure, you label it with these fluorophores that can be switched on and off. And I'm going to show you a movie that kind of demonstrates how this works. I'm going to use another structure in the cell as an example. These are microtubules. This is the other structure my lab is very interested in. And microtubules are the highways of the cell. They're about 30 nanometer polymers also. And they form this network in the cell that is very dense. You have many microtubules in close proximity. And when you take a normal light microscope image, it appears very blurry. Again, you can't really tell what this looks like. Where is one microtubule start and the other one end? But the trick in super resolution microscopy is to use these labels that can be switched on and off. So if I play this movie, you'll see these flashing lights, right? It's the same image where we have turned off the fluorophores, most of them, and we only activate a few fluorophores at a time. So those bluish reddish blobs that you see are individual fluorophores that are switching on and off. Every time one comes on, we identify it and we pinpoint its position and we put a dot to represent the position. And over time, as we collect more fluorophore positions and put more dots, we can start reconstructing these linear structures that are the microtubules. And we can now start discriminating their width and multiple ones that are in close proximity. Sorry, the movie stopped playing, but I hope you get the idea. Does that make sense to everyone? Questions?
So, okay, so I understand like turning one on and off, but are you still like, is it still like a light in like a large area, or is it like focused light on each?
No, so this is what is known as wide field imaging, meaning that we are capturing light from a wide field of view. Basically, the illumination, the light that we are putting into the microscope is illuminating a large area. And then we're collecting photons from that large area and imaging them on a CCD camera, right? A very sensitive CCD camera. And so this is just a kind of a zoom in to one part of the CCD camera. And each one of these little flashes of light is an individual fluorophore in that area that is switching on and off. Make sense? Yeah.
The question I have, are the fluorophores the same wavelength?
Mm-hmm. Yeah, so the fluorophores are the same wavelength. And we don't really have much control over which one we turn on and which one we turn off at any given point. So it's stochastic. We hit them with light, right? One wavelength of light puts them into a dark state. The other wavelength of light activates them into the bright state. The trick is, if you hit them with a lot of light, right, they will all be activated and all will be fluorescent. But if you hit them with very low light levels, stochastically, some of them will get activated, but most of them will not, right? And this allows you to get the sparseness. So you're only activating a few of them. They're sparse and they're not next to each other. The chances of the two of them being right next to each other and getting activated is pretty low. Does that make sense? Yeah.
Good question. Because our school has STAD.
STAD, yes, that works differently.
Yeah. Does storm also, so does it also photo bleach the particular what you're looking at? Because like STAD you can only visualize it once and then it's
So
non-fluorescent, is it?
that is a scanning technique.
Yeah.
You scan over the region with these donut illumination.
Yeah.
And it is pretty high intensity, so you do get photo bleaching.
Yeah.
In storm, you actually cannot get any information from one shot, right? You have to accumulate many images, right? Each frame only has partial information about where part of the fluorophores are. So you need to keep imaging for a while.
Oh, okay.
And eventually everything will get photo bleached. But
so
before they do, you locate their positions, right? And then you reconstruct a map of their positions, and that's your end.
Okay, yeah, that
Yeah.
might be better.
But yeah, I didn't talk about that because that's not what we do in my lab, but it's slightly different idea. Any other questions? Okay. So Can we now go back to this problem of the chromatin and visualize it using these high resolution imaging technologies where, I said we improved the resolution, but I didn't quite say how much. In fact, we can improve it by tenfold. So we can get from 200 to 300 nanometers to about 20-30 nanometers. So now that's getting more comparable to the size range of this chromatin fibers that we're interested in visualizing, right? And so this is what we did in my lab. So we can take that image and convert it into a super resolved image. And I hope you can see the transition from a blurry image to something much sharper and much more detailed. We now start seeing these more dotty structures, almost like these beads on a string like thing that the textbook model is telling us about. Yeah.
I have two questions.
Yeah.
So how long does it take for you to get
Get
this
that
image?
picture. Yeah, so it depends on what you're imaging and how many fluorophores you have to localize. It's a slow technique. So one image typically takes anywhere between 15 minutes to a 20-minute time period. Yeah, roughly. Yeah.
My second
So this is fixed cells. Sorry, I forgot to say that. It's
okay.
not living cells. They have been frozen in space.
Got it. And then my second question, you mentioned that the illumination sort of happens stochastically. How do you sort of, is it like a mathematical determination, or how do you determine at what point you stop that process?
The imaging, yeah.
Yeah.
So you typically keep imaging until there's no more signal and everything has photo bleached, basically. Yeah,
Okay, got it.
yeah. Just to ensure that you have acquired, the position of majority of the fluorophores. Yeah.
Okay, thank you.
Other questions? These are great. Keep them coming. Okay, so I'm not gonna go into the details of how we make sense of these images. It's actually very complicated mathematically. Once you get the points, these are not your typical images that you are used to in light microscopy. They're just what you get at the end of the imaging is a list of X, Y, and sometimes Z positions if you're doing 3D imaging. So what you're working with is just a list of points. And we have to rethink how we process that data, how we make sense of it, how we analyze it. I'm not going to go into the details of that, but we have, and this is just a zoom in of the region, just to show you the difference in resolution. But we have developed ways of processing this data, converting it from something like this to something like what you see here, where we are segmenting out different sort of structures and patterns in the images. One pattern you may notice is this green domain at the nuclear periphery. And that's heterochromatin. We can actually really nicely segment it out from the rest because it's denser. And this is reassuring to us because we know that from electron microscopy that condensed heterochromatin is associated to the nuclear lamina, to the edge of the nucleus. And this is what we see in our images as well. It's mostly at the nuclear lamina, and we see these invaginations into the nuclear interior, which really resemble the electron microscopy images. But we can sort of zoom into these domains that we segment. We notice that they're made up of smaller units, and we can go in and segment those smaller units as well individually. And then we can sort of calculate what each individual unit corresponds to in terms of how many nucleosomes are in there, etc. We do very quantitative analysis. Lo and behold, after doing all that, we came to this sort of new model of how chromatin is folded and organized inside the nucleus. It's more disordered than the textbook model. We don't only have like a 10 nanometer and a 30 nanometer fiber, but we have something much more heterogeneous. We have these units of compaction where multiple nucleosomes compact together, like in the textbook model, but this doesn't look like a typical 30 nanometer fiber. And we can have very varying numbers of nucleosomes in there. It's very heterogeneous. We call these nucleosome clutches, like egg clutches. And they can be, anywhere from 20 to 100 nanometers in size, depending on how many nucleosomes are assembling together to form these clutches. So in genomic terms, about 2 to 10 kilobases. And these clutches are separated by nucleosome poor regions, and they cluster together into these multiple domains. And so this is sort of more disordered than what the textbook model suggests, and much more heterogeneous. And, with multiple other technologies now, including cryo-electron microscopy, cryo-electron tomography, we're really coming to this understanding that inside the intact space of a nucleus, we don't have a 30 nanometer fiber. It's much more disordered. We have these multiple nucleosomes forming heterogeneous groups and domains. But still, we do have this more compacted and more open regions that can interconvert between each other to activate and repress genes. And we think this makes sense because the inside space of a nucleus is much more active, right? You have transcriptional machinery working. And this provides a more flexible template. You can more easily open something like this compared to a very highly compacted and organized 30 nanometer fiber. So again, why does this matter? Why do we care? This is a mechanobiology boot camp, so I thought I would give you an example from mechanobiology. Now that we can visualize this structure, we can also visualize how this structure is remodeled. And how am I doing in time?
You're fine, you have about 15 minutes. to go.
Okay, I
Yeah.
should be able to finish.
Yeah.
My lab teamed up with multiple people from CEMB, Suchin Hyo, Rob Mock, Vivek Shenoy, Jason Burdick, to understand how mechanical cues talk to the nucleus to change the structure, this organization, and remodel it and modulate it. Now, probably at this point, you know that the mechanics of a tissue is important for the cells that are within that tissue, and we can have very varying mechanics of different tissues in our body. Bone is much stiffer than brain. And from many years of work, we know that the nucleus is not isolated from its outside environment. It's actually tightly coupled. So on the nuclear membrane, maybe Ben talked about this, but we have the link complex. And the link complex gets connected to the actin and the microtubule cytoskeleton. So when the cytoskeletal contractility changes in response to mechanical cues, these forces are transmitted to the nucleus, and they can change, the nuclear sort of mechanics, first of all, but also how the chromatin is spatially organized inside the nucleus. And we knew that, for example, if you take a stem cell and put it on a soft substrate or a stiff substrate and give it differentiation cues, it can, on the soft substrate, differentiate into a lineage that is more resembling something you find in the body on soft tissues. Versus if you give it differentiation cues on a stiff substrate, it differentiates more into a cell type that you find on stiff tissues. And so how does the cell know, which direction to go? It knows because it turns on certain genes and turns off other genes. And that is controlled again by all this epigenetic regulation and the spatial organization of chromatin. So we wondered, how much does this organization change in response to mechanical cues? So to look at this, we took different substrates of different stiffness, particularly these stiff and soft hydrogel gels, and we took mesenchymal stem cells and grew them on these hydrogels, and then we used this very high resolution, super resolution microscopy method to visualize chromatin. Now these images are color coded from blue to red being the more compact, those heterochromatic regions of chromatin, and blue, the more open regions of chromatin. And I hope you can see that chromatin looks very different on different substrates, right? When the cells are on different substrates. So on the soft substrate particularly, you have a lot of lamina-associated chromatin at the nuclear lamina. That is very highly compacted. We think of this as the silenced compartment. So indeed, mechanical cues talk to the nucleus and remodel the spatial organization of the chromatin and control how much heterochromatin and how much euchromatin compartment you have inside the cell. And why does this matter? Well, if we take now cells that reside within a tissue in the body, so we took in this case tenocytes, which are tendon cells. So tendon is a very mechanically active tissue. It transmits forces between muscle and bone. Now, if we take these tenocytes from a healthy individual and visualize their chromatin and compare it to the chromatin of tenocytes that we took from a tendonitis patient, where because of inflammation, the tissue has become softer, and visualize the chromatin and compare it, we find that on the chromatin of the tenocytes coming from the tendonitis patient resemble these stem cells that were on the soft gel. So this is also happening in the body, where changes to the mechanical properties of the tissue due to injury, due to disease, are changing the spatial organization of chromatin in these tissue resident cells, and likely impacting that gene activity, what genes are expressed, and changing the phenotype of these cells. And then if we take, these healthy tenocytes and grow them on soft gels, we see that indeed they can now resemble a diseased tenocyte. So their chromatin is very pliable and can be remodeled in response to mechanical cues. I think I will stop there and sort of go back to Sidney Brenner's quote. So if I gave you one message in this lecture, let it be that new technologies really drive new biology. And, as you're going through your PhD and choosing your projects, be on the lookout for these revolutionary technologies and make use of them, because it's really an exciting time now with all this new developments in many different technologies. This is my lab, my collaborators, our funding. Happy to take any more questions. Yeah.
Thank you. The question I have is, does the lineage of the cell ab initio have an effect on how it reacts to the stiffness of the hydrogel? Because tendinocytes are from probably high pressure areas. If you take, for example, an adipocyte and put on the soft surface, are you going to see the same result, or is it going to be different?
That's a great question. I think it's going to be different. It's going to be cell type dependent. It's going to be dependent on whether it's a contractile cell or not. It's going to be dependent on the epigenetic state and the chromatin state, the starting state of that cell. And it's going to be different for an adipocyte compared to a tenocyte, certainly. So I think it's going to be very context dependent and very cell type dependent. Yeah.
Follow up to that. For the cells you used, the primary cells from patient. Do you think if you culture the cells on the culture plate before you put them on the different states, it's going to change the result?
Yeah, absolutely. I mean, I think this is important, right? We know, for example, we've done this with chondrocytes. And if you take them at passage 0, where they haven't seen any surface, versus passage 6, where you've passaged them on plastic for a while, they do have very different looking chromatin. So they sort of, the environment changes the chromatin of these cells. So they do adapt to, whatever environment they're growing on, and after a certain number of passages. So the question is, how pliable is that? Like if you now take passage 6 and put it back, or how long does it take before it becomes irreversible? And it's like, they lost the memory of what they were and where they came from. Those are really interesting questions. Yeah.
Thank you.
So how we were mentioning, of course, that like the actin is like connected to the nucleus and like it's a really like dynamic system. I think With the processes you're using to like look at like the genome and the nucleus, could we do like also imaging where we're also able to see the actin on the outside?
Oh, yeah.
Or
Yeah,
is that like a photo bleaching issue or something?
yeah, no, so you certainly can, this is another beauty of light microscopy, right? You have multicolor imaging, which you can't do with electron microscopy, right? So you can put different labels on different structures and visualize them in different colors. And you know, again, what you're looking at because you have labeled it. So you can certainly do something like that. We haven't done it with actin, but we have done it with other. So for example, trying to visualize chromatin at the same time as all other cell markers to see how the phenotype of the cell is changing. What markers is it expressing? Are those changing as the chromatin is changing? We can visualize RNA as well and look at whether a gene is active, it's producing mRNA or silenced as these structures change. Yeah, certainly.
And there's no issue of like, if when you were looking at like other markers, like it wasn't like an issue of it like blocking the nucleus when you were trying to also look at the genome or anything like that? Like, is there like physical?
I mean, there's definitely a lot of things you have to take into consideration when you're labeling multiple things together, right? So if you're labeling proteins, proteins in the nucleus versus like, you know, actin or other proteins in the cell, that's usually straightforward. You do immunofluorescence labeling, you use antibodies and antibodies from two different species that bind different things. And those protocols are often easy to combine together. If you're visualizing, let's say, a protein and RNA or a protein and some other nucleic acid, the protocols are different, so combining them becomes more challenging and harder. But people have worked out some of those details, so there's definitely ways of doing it, but you have to be careful and, you know, not to perturb or try to preserve as well as possible the structure that you're trying to visualize. Absolutely. Yeah. Yeah.
Can you render a 3D volume using Storm?
Yes. So I didn't talk about how you do that. What I showed you was 2D imaging where you have blobs, right? And you pinpoint in X and Y, their location. But you can do a trick. So what you want to know is when those blobs are out of focus, right? How far are they from your focal plane? And measure in Z, like whether they're up or down. And in fact, when those blobs are above or below the focal plane, they defocus. So their size becomes slightly larger. And so the size tells you where they are in the Z position. But the issue is above and below is symmetric. So you can't differentiate if a fluorophore is up or down. And to do that, what you do is you put a cylindrical lens into the imaging path, and the cylindrical lens deforms the image, makes it, introduces astigmatism. So that if the fluorophore is above the focal plane, it stretches in one direction, it becomes elliptical in one direction, and if it's below the focal plane, it becomes elliptical in the other direction. And the ellipticity tells you the Z position. So there is a way to do that so that you can map not just the XY position, which is the center, but also the Z position, which is how deformed the point spread function is. Yeah.
Thank you.
Yeah.
You mentioned that taking an image would take about like 15 to 20 minutes. I guess what is the current barrier to make it faster?
Making it faster. Great question. So it's how fast you can switch the fluorophores on and off. You're limited by basically the on switching is pretty fast. It's instantaneous. The off switching takes longer. Right now, each frame is like 10 milliseconds, 15 milliseconds. Because that's the time that it takes for a fluorophore to fully switch off so that you can switch on a new set of fluorophores. Because if you're imaging the same fluorophore in multiple frames, you're not really gaining new information, right? You need to switch them off and get the other ones to switch on. So that off switching is the limitation. There are ways of making that faster by hitting it with really a lot of light, but then that leads to photo bleaching. So there are, limitations. So I think what we're limiting to by in this field right now is how good our fluorophores. And people are working on developing new photo switchable fluorophores that switch on off faster using low light levels, etc. Yeah.
Is it, I guess a follow-up question, is it possible if you like, because we have so many different colors, what if you throw in a mix and excite different lasers?
Yeah, people have done that. You know, you can do up to maybe 4 colors. Beyond that, spectra start mixing, etc. So that gets you so far, but doesn't really like fully overcome this limit. But that's a great idea, and it's actually been done. Yeah.
So is there a technique where you could select the fluorophores that you want to come at a particular time? Like rather than be stochastic, you like,
So
all
that's-
these sets come out this set.
That's kind of the principle of STAD, this other method that we talked about. So what STAD does, and maybe I can draw it on here. STAD is a scanning technique, okay? So it's not just wide field illumination where you illuminate a lot of things, but it's scanning where you illuminate a very small volume. So you focus your light beam. When you focus your light beam, right, it's going to make a focused sort of spot that it illuminates, and it only illuminates things that are in that focused spot. But that's also limited by diffraction. How much you can focus your light is limited by diffraction. So you also get a volume typically that you're illuminating that is 300 or so nanometers. Now what STAT does is it overlays that focus excitation beam with another beam that is shaped like a donut, okay? So there is intensity here, and there is 0 intensity in the center. And where you have the intensity is where you switch off the fluorophores. So you turn these fluorophores off, and you only allow fluorescent light to come from the center of the donut. And that is much smaller. That's again like 10, 20 nanometers, right? So this is how you do STAD. Then you take that excitation and donut beam and you scan it across your sample. And so it's more targeted. You know exactly what you're switching off. Yeah. And it has advantages and disadvantages as well. You know, it takes more light. It's more damaging. But it's an alternative way of doing super resolution. Yeah.
We had a speaker yesterday sort of talk about nuclear
Mhm.
stuff. And so he also mentioned that, I guess, when a cell interacts with the substrate, it could be, the nucleus can be affected by actin directly interacting with the nucleus, or maybe the substrate cell interaction causes like a signaling.
Yeah, mechanical
Or
transduction.
the
Yeah.
chromatin sort
Which
of,
one is it?
like, what do you speculate as
Yeah.
far as your lab investigating?
It's a good question. It's a fantastic question. So if we do ablate the actin cytoskeleton, we no longer see the remodeling. So it's definitely actin contractility dependent, but that doesn't exclude other mechanism signaling. We haven't investigated that. It could be a combination. But yeah,
Thank you.
absolutely. Yeah. Yeah.
Just curious. So you're doing this on fixed cells, of course, right? And they were in like a 2D area. Could you do this with like cells that are like dividing? Like if you fix the cells and you were like looking for cells in different like division stages, could you like look at them and just see like how the chromatin like either like condenses even more? Or,
Yeah.
like,
Yeah, people have tried that with dividing cells. You can actually synchronize cells so that they all divide at the same
Really?
time, such that you can get many. Because otherwise, like to find one in a dish is like looking for a needle in a haystack. But if you synchronize them, like you can block microtubules, and then you can give a pulse, and then they all start dividing all at the same time, then you increase your chances of having dividing cells. And people have done that. The conclusions of those studies were that the chromatin of the chromosomes, the condensed chromosomes, is very similar. You still don't have 30 nanometer fibers. It's all these 10 nanometer fibers that are compacted. They become more compacted, but it's still pretty heterogeneous, and it still doesn't look like a 30 nanometer fiber. Yeah.
In the picture that you have with multiple colors, how do you differentiate regions, especially like in the center, you just have like multiple regions and blobs?
Oh, that segmentation I showed you, yeah, so this takes into account the points and how close the points are together. And we set a threshold and say, if the points are within a certain distance of each other, we will consider them to be part of the same structure. And if something is further apart, then that's sort of like the start of a new structure. So based on the proximity and the density of the points, we can set thresholds to segment things that way. Yeah. Lots of great questions.
Sufficient. So can you also add markers that go to specific genes or like
Yes.
just to know like exactly like what chromosome that is or something like that?
Great question. Yeah, we've done that and we're doing more and more on that. Because yeah, what I showed you was just chromatin, very global and non-specific. We don't know where each gene is, but we can use a method called fluorescence in situ hybridization, FISH. In this case, DNA FISH. And we can put our photo-switchable fluorophores on those FISH probes, and we can light up specific genes. Usually they look like a blob in your normal microscope image, but if you use these methods, you can actually see how far extended out it is or how compacted it is, what its volume in 3D space, etc. Yeah. And people have even expanded this concept to what is known as sequential FISH. So instead of labeling with many FISH probes and just looking at a large like megabase region, you can use shorter probes. And then you light up one probe at a time, right? So first you light up the first, let's say, I don't know, 10 kb segment of the gene, and then you light up the next 10 kb and the next 10 kb, etc. So you are actually walking along the gene so that you can see how it's sort of folded in the 3D space of the genome. So again, this concept has been used to really map out with very high spatial resolution how different sequences of the genome contact each other, where they are in space. Yeah, so it's been quite an exciting field.